So today I'm going to discuss carbohydrate metabolism with you. First of all, we'll come to the glucose metabolism. So just to inform you that all the figures and the diagrams that has been added in this lecture are taken from Lippincott. However, the other material is taken from the different sites. So this is a picture which shows role of glucose and many other carbohydrates and their presence in the different cycles. So first of all, we will discuss glucose metabolism, how glucose is broken down into through the process of glycolysis and Krebs cycle, and then how glucose is resynthesized, that is gluconeogenesis. Then we will go to the fructose metabolism. Then we will go to the galactose metabolism, glycogen metabolism, and pentose phosphate shunt, etc. So this is the recap of all the cycles that are taking place in the body simultaneously. So first of all, glycolysis. Glycolysis is the process through which glucose, which is 6-carbon compound, it is broken down into 3-carbon pyruvate. This process of glycolysis takes place in the cytoplasm. Now, glucose is broken down to glucose, not broken down, but synthesized into glucose 6-phosphate in the presence of the enzyme glucokinase or hexokinase. In this process, one phosphate is required to make glucose 6-phosphate, and for this, one ATP is broken down. So here energy is utilized. This reaction, it requires energy. So ATP, one ATP is broken down into one ADP, and the phosphate, the remaining phosphate, it goes to the glucose to form glucose 6-phosphate. Now these two enzymes are very important, which are glucokinase and hexokinase. As you can see the table on the right side, glucokinase is present in liver and some of the beta islet cells of the pancreas. This glucokinase has a high Km and high Vmax value. It is insulin sensitive. As you can see, it is present in the beta islet cells of pancreas. So definitely beta islet cells of pancreas also secrete insulin. It means that this glucokinase is associated with the insulin secretion from the pancreas. Now, glucokinase enables glucose entry into the liver, and hence it further metabolism through the glycolysis process. So if there's more glucose in the body, beta islet cells are also going to secrete glucokinase, and this glucokinase eventually helps in the mobility of the glucose into the liver. Therefore, as it is also glucokinase is also secreted by some of the beta islet cells of pancreas, glucokinase is insulin sensitive. Therefore, if there is more insulin, glucokinase will enable the glucokinase, more glucokinase will be there, and hence glucose entry will take place in the liver. If there is any mutation in the pancreatic beta islet cells genes, then it lead to a disease which is known as Moody type 2, which is maturity onset diabetes type 2. So this happens because of the altered secretion of the glucokinase by the pancreatic cells, and hence any mutation can lead to Moody type 2. The other enzyme in the conversion of glucose to glucose 6-phosphate is hexokinase. So this hexokinase is present in the extrahepatic tissue. It can be present in skeletal muscle, it can be present anywhere in the body, but not in the liver. Hexokinase has decreased Km and decreased Vmax value. Hence, it enables the extrahepatic tissue to uptake glucose and convert into glucose 6-phosphate. Now, glucose phosphate is a high energy bond. So this, when it's converted into glucose 6-phosphate, the glucose, after conversion to glucose 6-phosphate, it gets trapped into the cell. Once it's trapped into the cell, it is eventually yielded to glycolysis process. Therefore, limited glucose is converted to glucose 6-phosphate. That means that the cells, the extrahepatic cells, will convert that much amount of glucose into glucose 6-phosphate, which is required for them. not all of the glucose that is available. So rest of the glucose can be sent out of the blood, and this is then this is due to the, sorry, this is a typographical error, id likha hua hai. This is due to the less Vmax and Km value of the hexokinase. As you can see that hexokinase convert glucose to glucose 6-phosphate, that it is regulated by glucose 6-phosphate present into the cell. If there is more glucose 6-phosphate into the cell, more of the glucose is being trapped into the cell, that less of the hexokinase will be available. Hexokinase will be auto-regulated by the glucose 6-phosphate presence. Then glucose 6-phosphate is converted into fructose 6-phosphate. So it's very interesting here. As you can see, there are two arrows. So one is going downward from the glucose to glucose 6-phosphate, while the other is going upwards, which means that the fructose 6-phosphate can be converted back to glucose 6-phosphate, or glucose 6-phosphate can be converted back to fructose 6-phosphate. So they can be interchangeable. However, this glucose to glucose 6-phosphate is just one arrow, single arrow, directed downwards. That means that cannot be converted. Glucose 6-phosphate cannot be converted back to glucose unless we utilize some other enzyme that can overcome this hurdle of the ATP which was being consumed here. So we are going to describe this in detail in the next slides. Now, synthesis of fructose 6-phosphate into fructose 1,6-bisphosphate. So again, you can see here over fructose 6-phosphate, there was just one phosphate, but now we have bisphosphate. Two phosphates are being added to it, and again, due to the breakdown of the ATP into the ADP. So one phosphate from the ATP is gone to the fructose to form fructose 1,6-bisphosphate. The enzyme that is required in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate is phosphofructokinase 1, also known as PFK1. Now, this PFK1 is very important. Why it's important? Because this is the most regulated enzyme of the process of glycolysis. It is also the rate limiting step of glycolysis. It is so by having said so that there's a rate limiting and the more major regulator, so the process of glycolysis is regulated in other terms by phosphofructokinase 1. Now, what are the regulatory molecules that regulate phosphofructokinase 1? So there are positive regulators and there are negative regulators. So positive regulator is AMP or cyclic AMP, which means the less energy in the body. If there's less energy in the body, they can cause the activation of phosphofructokinase 1. So if phosphofructokinase 1 will kick in, it will enable the conversion of fructose 1,6 or fructose 6-phosphate into fructose 1,6-phosphate, and hence the process of glycolysis will take place and will move forward. I'm repeating it again that a phosphofructose kinase 1 PFK1 is very important. You can see it's a unidirectional step where it cannot convert back unless we utilize the other enzymes. So the positive regulator is low energy. Cyclic AMP or AMP, adenosine monophosphate, low energy in the skeletal muscle or the extrahepatic tissue, is the positive regulator of phosphofructokinase 1. If it's low energy, we all know glucagon will kick in and hence phosphofructokinase will be activated. Then fructose 2,6-bisphosphate is also a positive regulator of fructokinase 1 in the liver. So in the skeletal muscle is cyclic AMP and AMP, which is low energy, or in the extrahepatic tissue, while in the liver, fructose 2,6-bisphosphate is a positive regulator of the phosphofructokinase 1. So the negative regulators ATP. So when there is ATP, there is no need to break down of glucose and there is no need of glycolysis, and hence phosphofructokinase 1 will not be activated. It's been negatively regulated by ATP. And again, citrate is the negative regulator. Now, fructose 1,6-bisphosphate is converted into two compounds. Up till fructose 1,6-bisphosphate, it is 6-carbon compound. Now, this 6-carbon compound is broken down into 3-carbon, which is either dihydroacetone phosphate or glyceraldehyde 3-phosphate. So dihydroacetone phosphate, DHAP, this is also 3-carbon. This is also 3 carbon. And glyceraldehyde 3-phosphate. This is also 3 carbon. Now, dihydroacetone phosphate readily converts into glyceraldehyde 3-phosphate by the presence of enzyme triose isomerase. This is triose because this is 3 carbon. So it's 3 carbon converts easily into 3 carbon. So it's bidirectional. Glyceraldehyde 3-phosphate can be converted into DHAP, dihydroacetone phosphate, or dihydroacetone phosphate can be converted into glyceraldehyde 3-phosphate. Now, the important thing is this, that for the process of glycolysis, glyceraldehyde 3-phosphate is important. So DHAP will convert into glyceraldehyde 3-phosphate, which will further take the lead towards the synthesis of pyruvate. This is 3 carbon. Glyceraldehyde 3-phosphate is 3 carbon compound. Thus, two cycles, not cycles, but two reactions simultaneously from glyceraldehyde, one glyceraldehyde and then the other glyceraldehyde. So two glyceraldehyde will simultaneously go down in the process of glycolysis to produce two pyruvate, which is two pyruvates, which are the 3 carbon. So glucose was 6 carbon, glyceraldehyde 3-phosphate is 3 carbon. So 3 plus 3 makes 6. It means that the glyceraldehyde, two glyceraldehydes will simultaneously take their part in the process of glycolysis. Now the glyceraldehyde 3-phosphate is converted into 1,3-bis phosphoglycerate. This is done by the this is done by the presence of NAD, which is converted into NADH and H positive ion. Along with that, the enzyme which is required is glyceraldehyde 3-phosphodehydrogenase and the inorganic phosphate. So the inorganic phosphate is then entered into the enabled the formation of 1,3-bis phosphoglycerate. 1,3-bis phosphate can also be converted back into dihydroxyacetone phosphate or glyceraldehyde 3-phosphate. So the reaction, this reaction is again conversion of these two into 1,3-bis phosphoglycerate is two-way. Now 1,3-bis phosphoglycerate is converted into 3-phosphoglycerate. With the synthesis of 3-phosphoglycerate in the presence of enzyme, phosphoglycerate kinase 1-ATP is produced. Why? Because it was 1,3-bis phosphoglycerate. So one phosphate on carbon 1 was removed, and hence just 3-phosphoglycerate was left behind. So this, not carbon, phosphate on the first carbon was removed. So this phosphate helps in the synthesis of ATP. Now this is the substrate level phosphorylation. And this formation of 3-phosphoglycerate with generation of 1 ATP, this is the first substrate level phosphorylation of glycolysis. Then the 3-phosphoglycerate, it is converted into 2-phosphoglycerate. The enzyme which is required is phosphoglycerate mutase. They can also be interconverted. However, I forgot to put the other arrow, but they can also be interconverted. And then 2-phosphoglycerate in the presence of enolase is converted into phosphoenol pyruvate. Now this enolase in the enzyme, it is the this enzyme causes dehydration. So dehydration means it brings out water. So once this water is brought out, the product that is left behind is phosphoenol pyruvate. This phosphoenol pyruvate is then converted into pyruvate in the presence of the kinase enzyme. You can also say pyruvate kinase, and again 1 ATP is produced. This ATP, where phosphoenol pyruvate is converted into pyruvate and the phosphate group is left behind, this phosphate group enters into ADP to form ATP. Now, this formation of ATP is the second substrate level phosphorylation in the process of glycolysis. So this is very important that there are two substrate level phosphorylation, the glycolysis. So one was here, which is 1,3-bis phosphoglycerate to 3-phosphoglycerate. This was the first phosphor because 1 ATP was produced in the reaction. So this was the first substrate level phosphorylation. And the second substrate phosphorylation, ATP is produced by the phosphoenol pyruvate into conversion to pyruvate, and 1 ATP is produced. Now again, as I told you that from glyceraldehyde 3-phosphate, two reactions, 2-glyceraldehyde 3-phosphate will simultaneously go down to form eventually 2-pyruvate. So 2 ATP plus 2 ATP will be produced. 2 ATP in this, 1 ATP from one. Here just 1 ATP and then 1 ATP from the another glyceraldehyde. Again, here 1 ATP in this reaction and 1 ATP in the other reaction. So we are going to get 4 ATPs at the end, and over on the top, as we can see here, the 2 ATPs were utilized. So overall, we had the net gain of 2 ATPs. Now, enolase kya karta hai? So what is the importance of the enolase enzyme? So this is medically very important because enolase is present into the toothpaste and into the mouthwash. What is the role of enolase? Is it inhibited by fluoride? So the fluoride, which is fluoride in the toothpaste, this enolase is inhibited by fluoride because enolase is not present in the toothpaste. Fluoride is present to the toothpaste. So our toothpaste and mouthwash, they contains fluoride. So fluoride inhibits enolase, and what it does, that the bacterial enolase is inhibited by the fluoride present into the toothpaste or the mouthwash. Once bacterial enolase is inhibited, then the process of glycolysis in bacteria which are present on our teeth will not take place. And by doing so, the dental caries or the dental plaques can be alleviated or can be inhibited. So up till now, this is 3-phosphate, right? Now, what are the characteristics of glycolysis? So the characteristics of glycolysis is that the 6-carbon glucose is converted into 2 pyruvates, which are 3 carbon. So 2 reactions will take place. I'm repeating it again and again because 2 reactions will take place after DHAP, 3 carbon or 3 glyceraldehyde 3-phosphate, so 3 carbon. So DHAP can move forward, but DHAP is eventually converted to glyceraldehyde 3-phosphate in the process of to take place in the process of glycolysis to synthesize pyruvate. So two steps, 2 reactions of this thing after glyceraldehyde 3-phosphate 3 carbon will take place. Now the irreversible reactions. The irreversible reactions are the one that cannot be where the products cannot be converted back to their substrate. So there are 3 irreversible reactions which are overcome during the glycogenesis through the process of different enzymes, through the presence of or through the induction of different enzymes. We will also discuss in the last, but to mention you the important steps which were irreversible and the other enzymes were utilized. They are made pink in here. So glucose, when it was converted to glucose 6-phosphate by the conversion of 1 ATP into ATP, yani 1 utilization of ATP, this glucose 6-phosphate can be converted back to glucose by the induction of enzyme glucose 6-phosphatase. Likewise, fructose 6-phosphate into fructose 1,6-bisphosphate, which was because of the utility of 1 ATP was again converted, can be converted back into fructose 6-phosphate by again utility of the other enzyme, not phosphofructokinase, but by the fructose 1,6-bisphosphatase, because these enzymes can overcome that high energy level and can resynthesize that fructose 1,6-bisphosphate back to fructose 6-phosphate. Then the other one is the pyruvate. Pyruvate cannot convert back into phosphoenol pyruvate. So this pyruvate eventually first converts into oxaloacetate, and then oxaloacetate converts back to phosphoenol pyruvate. Now how pyruvate converts into oxaloacetate and then back to phosphoenol pyruvate, that I'm going to describe in detail for next slides. But over here, the pink ones are the one which are actually irreversible reaction, that were actually irreversible reaction, but by the incorporation of the separate enzymes which had the power to overcome the high energy, once they were employed, the substrate, the products were eventually able to convert back into their substrates. Then comes the link reaction. I hope you can see the one which is headings that are written here. So sorry. So what are the link reactions? So link reactions are the ones where the pyruvate is converted into acetyl-CoA. So pyruvate can enter into mitochondria, can be converted into acetyl-CoA by the presence of an enzyme complex, which is pyruvate dehydrogenase enzyme complex. Now, pyruvate dehydrogenase enzyme complex utilizes NAD and NADH plus H positive ion is produced, and one carbon dioxide is released. So it's dehydrogenase complex is dehydrogenases. It releases one carbon dioxide, it reduces NAD, gives it to makes it into NADH plus H positive, and results in the product formation of acetyl CoA. Now, interesting thing, pyruvate was 3 carbon. This pyruvate was 3 carbon, acetyl CoA is 2 carbon because 1 carbon is removed. Over here, you can see carbon dioxide comes out. This carbon dioxide will remove 1 carbon from the pyruvate, and hence the 2 carbon acetyl CoA is eventually yielded. Now, this is a link reaction. Why? Because this acetyl CoA will eventually go into the our citric acid cycle or TCA or Krebs cycle. So important thing, one more important thing that I want to discuss here is NAD. NAD is a high energy bond. It releases almost approximately 3 ATPs. So wherever NAD is converted to NADH plus H, 3 ATPs is formed. Likewise, FAD yields approximately 2 ATPs. So 2 ATPs for every reaction where you see one FAD is converted into FADH and H positive, 2 ATPs are formed. One GTP can lead to one GDP conversion to GTP can lead to is equal to 1 ATP. Likewise, 1 ADP conversion into 1 ATP is also equal to 1 ATP. So wherever you see these NADs, FADs, GTPs, and ATPs, please make this clear that NAD makes approximately 3 ATPs. FAD makes approximately 2 ATPs. So wherever these NADs, FADs are, you see them, you can easily calculate the formation of ATPs by yourself. And then FAD makes 2, ATP is just 1 ATP. Likewise, GTP is equivalent to 1 ATP. Now, so in the link reaction, coming back to our link reaction, pyruvate and then its conversion into acetyl CoA. So pyruvate is converted to acetyl CoA by the presence of pyruvate dehydrogenase complex. So this complex has 3 subunits, E1, E2, and 3. Mentioning of these subunits over here is just to make you know that all these 3 subunits, they together require 5 coenzymes. These 5 coenzymes catalyze decarboxylation of pyruvate to acetyl CoA. Decarboxylation means that it enables removal of 1 carbon from pyruvate to acetyl CoA. So once this carbon is removed 3-carbon pyruvate is converted into 2-carbon acetyl-CoA. Now, acetyl-CoA is the allosteric inhibitor of pyruvate dehydrogenase complex. So this acetyl-CoA also, if there is more acetyl-CoA, it will eventually inhibit pyruvate dehydrogenase complex itself. If it's less acetyl-CoA, then pyruvate dehydrogenase complex will more will be activated, or more will kick in the conversion of pyruvate into acetyl-CoA. Now, the importance of those 5 coenzymes. Those 5 coenzymes which were important for decarboxylation are: number 1, thiamine, pyrophosphate, which is the active form of thiamine, vitamin B1. NAD is a derivative of niacin, vitamin B3. FAD is derivative of riboflavin, then lipoic acid, and 5th coenzyme is pantothenic acid, vitamin B5. Hence, vitamin B complex is very important for this link reaction. Any deficiency of these vitamins, let it be vitamin B1, vitamin B2, riboflavin, B2, NAD, vitamin B3, B5, any deficiency of vitamin can halt this link reaction. So once, if there's no link reaction, no acetyl-CoA will be produced. So once no acetyl-CoA is produced, no Krebs cycle will take place. No formation of citric acid cycle will take place. We're not going to have all the reactions come. And then acetyl-CoA is very important. It just not only goes to citric acid cycle, it goes to our fatty acid synthesis as well. It goes to our ketone body synthesis as well. So acetyl-CoA is very important, and if it's not produced in the body, many of the major reactions in the body will be stopped. So that's why vitamins are important. Then we come to the, now before embarking on the citric acid cycle, let me tell you about the different fates of pyruvate. So one fate is that it can be converted to the link reaction to acetyl-CoA. So the other is conversion of pyruvate to lactate. So this pyruvate can be converted into lactate by the process of lactate dehydrogenase, which converts NADH to NAD. Now this NAD, from its reduced form, it is converted to its oxidized form, because the enzyme dehydrogenase enzyme, it is actually oxidizing it. Now this NAD is important. It is required. It can be used into the glyceraldehyde 3-phosphate reaction and glycolysis. It can go anywhere where NAD is required. And once NAD is converted back to NADH plus, and through the electron transport chain, it can yield approximately 3 ATPs. So oxidation of the NAD is important, and this is very important. Lactates generation also helps in the generation of NAD, making it available for glyceraldehyde 3-phosphate, so that more and more of the glycolysis process can take place, and more of the energy can be produced. Now TCA cycle, Krebs cycle, or citric acid cycle. So pyruvate from the cytoplasm can enter into the mitochondria and can start the TCA cycle by conversion to acetyl-CoA through the reaction, which was the link reaction. Acetyl-CoA can act as an inhibitor of pyruvate dehydrogenase complex. So one more thing that I'm sorry I forgot, that this pyruvate dehydrogenase complex can also be inhibited by arsenic. So if there is deficiency of vitamin, person cannot have this link reaction. And besides this, if there is arsenic poisoning, which is a metal, and if there's arsenic poisoning, pyruvate dehydrogenase complex can also be inhibited, and again no acetyl-CoA will be produced. Now acetyl-CoA act as an inhibitor of pyruvate dehydrogenase complex, that I already told you. While on the other hand, so acetyl-CoA has a positive effect of pyruvate carboxylase enzyme. So what is the importance of pyruvate carboxylase enzyme? The importance that it facilitates the conversion of pyruvate to oxaloacetate. Oxaloacetate, why? Because pyruvate is 3 carbon and oxaloacetate is 4 carbon. So first it was decarboxylated to acetyl-CoA, which is 2 carbon. Now acetyl-CoA can have a positive effect on pyruvate as well. Acetyl-CoA itself is not doing anything. It's not coming into the reaction, but it has a positive effect on pyruvate, which is 3 carbon. So this 3 carbon pyruvate can be converted through carboxylase enzyme into oxaloacetate, which is 4 carbon, thus making this reaction where 3 carbon pyruvate to the 4 carbon oxaloacetate, and 4 carbon oxaloacetate eventually combines with 2 carbon acetyl-CoA to form 6 carbon citrate, and hence again the same 6 carbon compound we eventually get. The cofactors and the coenzyme which are required for the conversion of pyruvate to oxaloacetate are carbon dioxide, ATP, biotin, and magnesium. Now, 4 carbon oxaloacetate combined with 2 carbon acetyl-CoA to make 6 carbon citrate. Now, this is why when citrate is formed, that's the reason this cycle is also known as citric acid cycle. The enzyme that is required for the conversion of oxaloacetate to acetyl-CoA is citrate synthetase. Now, this is also in the citric acid cycle, this is the most regulated enzyme. So once it's the most regulated enzyme, it helps in the maintenance and in the regulation of the citric acid cycle or the TCA cycle. Now, when citrate is formed, it has a negative effect on the citrate synthetase. So it inhibits more actions of the citrate synthetase. So if there's less citrate synthesis will allow more citrate formation, and hence the citric acid cycle will continue. However, if the citrate is more in quantity, citrate synthesis will be hampered, will be inhibited, and then less citrate will be formed. So less number of the citric acid cycles will take place. This is all depends upon the energy and the ATPs and the situation where we are at the moment where we require energy. For example, if we are in the fear or flight moments where there is epinephrine, norepinephrine or noradrenaline in our body, where there is excess of cortisol in our body, maybe in this fasting and starvation state where we have now the surge of glucagon in the body. So if more energy is required, then, or if, for example, we have postprandial, we have excess glucose in the body, then these auto-regulated the regulated enzyme, the regulatory enzyme, they enable the regulation and hence enable the formation of the ATPs, appropriate number of ATPs, so that appropriate energy be provided to the body, and the excess of the substrates may be go and stored in the body for the future use. Now, citric acid cycle. This is citric acid cycle where acetyl-CoA, which is C2, 2 carbon combined with oxaloacetate, 4 carbon to form 6 carbon citrate. Here. So we're here in the citric acid cycle, the importances of these enzymes, these NADs, and then the conversion of different carbons into the lesser numbers from 6 to 5 carbons, 4 carbons, and then eventually back to 6 carbon. Now this citrate is 6 carbon. Citrate is converted into isocitrate, which is also 6 carbon, excuse me, in the presence of enzyme aconitase. Now, importance of aconitase. I'm sorry, maybe you're not able to see this thing because of this bar here, but the top table is for the aconitase enzyme. Now, this aconitase enzyme is in the rat poisons. The rat poison is made in such a way that inhibits this aconitase enzyme. Rat poison basically contains fluoroacetate, which converts to fluorocitrate, and hence it inhibits TCA by inhibiting aconitase enzyme. So that's the reason that rat poison can be injurious to anyone, not only the rats, not only to the rodents, but also to anyone who can accidentally ingest them, because aconitase is inhibited. So once it is inhibited, isocitrate will not be formed, and the TCA will not be the reaction of the TCA will not be carried forward. Then we come to the alpha-ketoglutarate dehydrogenase complex. So alpha-keto, now this isocitrate is then converted into alpha-ketoglutarate, which is in the presence of isocitrate. Dehydrogenase, nothing much. One NAD is converted into NADH and H positive with the release of one carbon dioxide. So one with the release of one carbon dioxide, five carbon is left, which is alpha ketoglutarate, and NAD is converted into NADH plus H positive. So hence one NAD can form approximately 3 ATPs. So you can calculate from here now onwards कि कितने ATPs are produced, because 3 here, 3 here, 2 here, and 3 here, and 1 GTP, 1 here. And yet again, one more thing that I want to explain is this, that the acetyl CoA is produced from pyruvate. So 3 carbon pyruvate converted into acetyl CoA. So for the one glucose molecule, not only 2 pyruvates were produced, but also for one glucose molecule, 2 Krebs cycles will take place simultaneously. So jitne ATPs are being produced here by NADs and FADs and GTPs, they are all multiplied by 2. And that's by doing so you and one ATP is also converted here, where through the link reaction by 3 ATPs jahan pe NAD was converted into NADH and H plus, so again multiply by 2. So all these will be multiplied by 2, because 3 carbon reaction jo tha, it was carried out by the 3 carbon phosphate. So 2 citric acid cycle will also take place. So all the ATPs from NAD, approximately 3 ATPs are produced to 3, and then multiply by 2 for every NAD, 3, for FAD2, for GTP1, and then multiply by 2. So you're then, this is how you are going to calculate your ATP. For example, if an examiner asks you how many isocitrate ATPs are produced into alpha ketoglutarate, so for one NADP produce approximately 3 ATPs, and then of course for one glucose molecule 2 citric acid cycles will take place, to 2 3s are 6 ATPs will be produced. Now alpha ketoglutarate is the 5 carbon compound which is converted into 4 carbon succinyl CoA. Again here one NAD is converted into or reduced to NADH and H positive ion, and also a release of carbon dioxide, and alpha ketoglutarate dehydrogenase enzyme is utilized here. Now one carbon release will eventually make it 4 carbon succinyl CoA. Now over here alpha ketoglutarate dehydrogenase complex is important. This dehydrogenase enzyme is important to get mentioned here, because this complex, this alpha ketoglutarate dehydrogenase complex is very much similar to the pyruvate dehydrogenase complex. Now the pyruvate dehydrogenase complex, the way it required those 5 coenzymes, thiamine, asin, and with FADs, thiamine, NAD, lipoic acid, and vitamin B5. Likewise, those 5 coenzymes are also required here to activate this alpha ketodehydrogenase enzyme complex. And if any of those vitamins is missing, because all 5 coenzymes were important to activate the enzymes or to enable the enzyme to act properly. So if any of the vitamin is missing, the action of the enzyme will not be proper, and hence the proper desired result will not be attained. So these 5 coenzymes are important, and by in addition to that, as I told you that the pyruvate dehydrogenase complex was also inhibited by arsenic. Likewise, alpha ketoglutarate dehydrogenase complex can also be inhibited by arsenic. So arsenic poisoning not only hampers our link reaction, acetyl CoA precursor of upper Krebs cycle is not produced, but it can also, if any of the acetyl CoA through any other reaction is produced, let it be from the fatty synthesis, from maybe from the ketones or whatever, but or maybe taken in diet or whatever, alpha ketoglutarate over here, this enzyme alpha ketoglutarate dehydrogenase complex, that would be inhibited by vitamin B deficiency, B complex deficiency, or maybe by arsenic poisoning. And hence, again, the other ATPs which were supposed to produce by the citric acid cycle will not take place. So this is the importance of the alpha ketoglutarate. Now, the succinyl CoA is converted into succinate through the process of through the enzyme which is on thiokinase. Thioester link is broken down and 1 GTP is converted into GTP. So 1 GDP is converted into GTP. Now this GTP is equivalent to 1 ATP. 1 ATP production. Then succinate is 4 carbon. It is converted into fumarate. In here, FAD is converted into FADH. So one FAD approximately produces 2 ATPs, and hence again you can calculate the ATP synthesis. And fumarate can be converted to malate, and malate can be converted into back to oxaloacetate. But through this conversion to malate to oxaloacetate, again one NADH and NADH is produced, and H positive ion is produced, and hence again approximately 3 ATPs are produced here by one malate into one oxaloacetate. This malate can also move freely out of the mitochondria into the cytoplasm, come back, and then this malate is one of the important substance that helps in the synthesis of pyruvate, convert malate into oxaloacetate and back to pyruvate synthesis in the irreversible reactions of glycolysis, where pyruvate cannot be converted to phosphoenol pyruvate because malate can be converted to oxaloacetate. तो वहां से it can jump to form the phosphoenol pyruvate synthesis. Now, gluconeogenesis. Gluconeogenesis is a conversion of, so that was, before the slide was the breakdown of glucose eventually for the synthesis of energy. Over here is the synthesis of glucose from, or conversion of the non-carbohydrate sources to glucose. So there are non-carbohydrate sources are propanol CoA, alanine, glycerol, lactate. Propanol CoA is very simple. It is converted into methylmalonyl CoA, which converted into succinyl CoA. So succinyl CoA you can see here. Propanol CoA, methylmalonyl CoA, and then to succinyl CoA. Now, succinyl CoA is present to the citric acid cycle. That succinyl CoA from propanol CoA can enter into the citric acid cycle and then hence form fumarate, malate, oxaloacetate with the corresponding ATP synthesis. Then alanine and lactate can also be converted to pyruvate. And now we will talk about the irreversible steps in the glycolysis, which are overtaken by the other enzymes to form glucose. Now these are the ones. These are the ones where utilized the products. So the pink ones, as I already told you, कि ये यहाँ से तो directly phosphatase converts glucose 6-phosphate back to glucose, fructose 1,6-bisphosphate converted back to fructose 6-phosphate by the fructose 1,6-bisphosphatase enzyme because they can overcome the high energy. However, this pyruvate is a little tricky because this pyruvate cannot be converted back to phosphoenol pyruvate. So what happens? Pyruvate is first converted into oxaloacetate through carboxylase enzyme and then to malate. Now the malate can go into and come out, no malate can come out of the mitochondrial cytoplasm, converts back into oxaloacetate, and the oxaloacetate then in the presence of carboxykinase is converted to the phosphoenol pyruvate. So pyruvate interconversion into malate and oxaloacetate is important for the resynthesis of phosphoenol pyruvate. That's it, because pyruvate, it cannot convert back to phosphoenol pyruvate. So carboxykinase enzyme is important. First, carboxylase is important. So pyruvate is converted to oxaloacetate and then phosphoenol pyruvate carboxykinase to form phosphoenol pyruvate. And hence, then again, once phosphoenol pyruvate is produced, you can see from here, all reactions are reversible reactions. Now the glycogen synthesis. What happens when we have the excess glucose? So important concepts of glucose are done. Now, what is the fate of glucose and fructose? Because glucose is the major source. So glucose is most of the time it is utilized and it can be stored. So if there's excess glucose, what happens? So excess glucose is stored in the body in the form of glycogen. Glucose can be converted to glucose 6-phosphate, which can be readily, easily converted back to glucose 1-phosphate. Now, glucose 1-phosphate combines with the presence of transferase enzyme to UDP glucose. Now this UDP glucose forms glycogen. So the story behind synthesis of glycogen is this, that the UDP glucose along with the glycogen, which is a protein having a tyrosine component in it, this protein and UDP glucose will combine together. UDP will remove and the glucose glycogen is formed, which is the primer for the glycogen synthesis. More and more glucose, UDP glucose, will come into being and it attached to the glycogen, and hence the chain of glycogen is produced. So these chain is the straight chain. Now glycogen is a branch structure. It has straight chains and then it has the branches in it. So the straight chain has the bonds of alpha 1 4 glycosidic linkages, while the branches, where the branches start, those branching areas have the alpha 1 6 glycosidic linkages, and hence these linkages enable more and more of the glucose entry into it and it's being stored as the form of the glycogen. And this is important because glycogen is not only stored in liver but can also be stored in the extrahepatic tissue. Now the glycogen that is stored in the liver, its importance is this, that it can be utilized whenever in the fasting or the starvation state, and for the synthesis of glucose. Just a glucose, it is broken down, glycogen part is being on the fasting stage through the presence of the enzyme, which is glycogen phosphorylase. It is broken down to glucose 1-phosphate or simply glucose. It can be broken into glucose. Now this glucose can enter into the blood. So the glycogen that is present in the liver is only for the maintenance of the blood glucose level in the blood, in the circulation, and that is during the fasting or starvation states mostly. But what about the glycogen that is present in the extrahepatic tissue? For example, if it is present in our skeletal muscles. So if there's a starvation state or in a fasting state or where more energy is required, then again the glycogen phosphorylase enzyme can break this glycogen back to glucose or glucose 1-phosphate into the muscles or into the extrahepatic tissue wherever it is present, and that glucose will not go into the blood but will be utilized for maintenance of the integrity and health of that tissue, which is in the form of starvation or fasting. So this is important, that more glucose that is present in the body can be converted into glycogen. UDP glucose, glycogen forms the primers of the glycogen. More and more glucose will come into being. UDP remove hota jayega, glycogen it will form. It's a branched structure. So straight chain will be alpha 1 4 glycosidic, where the branches start 6 to 8 units afterwards forms the branches. So the branches will be alpha 1 6 glycosidic units. Whenever the glucose is required by the body, this glycogen is broken down. So glycogen can be stored anywhere in the body, in the liver and the extrahepatic tissue. For the liver one, it can give glucose to the blood to maintain the blood glucose level. But for the extrahepatic tissue, that is only for the maintenance of the integrity and the function of that extrahepatic tissue, because that extrahepatic tissue also needs survival during starvation and fasting state or whenever the energy is required. So glycogen phosphorylase enzyme will come, it severs one of the glucose molecule and gives that glucose molecule for the utilization. So that's the story of the glycogen. Then we come to the pentose phosphate pathway. So pentose phosphate pathway, over here it is also known as monophosphate hexose monophosphate shunt. Pentose phosphate pathway is very important because it helps in the synthesis of purines and pyrimidine. It helps in the synthesis of the backbone of our DNA, adenine, cytosine, guanine, thiamine. So हमारे जो purines and pyrimidines which are present in our body, they are formed by this pentose phosphate pathways. Over here, if I say glucose, so you can see glucose is also a part of this reaction. This glucose can be converted into glucose 6-phosphate, goes to form gluconolactone, gluconate, converted into ribulose 5-phosphate. You can simply say glucose converts into glucose 6-phosphate, and glucose 6-phosphate can help in the synthesis of ribulose 5-phosphate. Now this ribulose 5-phosphate forms ribose 5-phosphate because we have ribose sugar, we have deoxyribonucleic acid or ribonucleic acid, DNA and RNA. So this ribose, this 5-phosphate is important. This ribose will eventually convert into phosphoribosyl pyrophosphate, phosphoribosyl pyrophosphate, PRPP, which helps in the synthesis of purines and pyrimidines. The intermediates of these are xylulose, which is N-acetyl-heptulose, erythrose, and glyceraldehyde, etc. So glyceraldehyde is not an intermediate, but you can see that glucose hai, to glucose can be converted into fructose, fructose can be converted into glyceraldehyde 3-phosphate. Now this glyceraldehyde 3-phosphate can either go into the process of glycolysis or can go back into the synthesis of the ribose with the formation of the intermediates. But you can see if we take these part, just glucose, so glucose convert into glucose 6-phosphate. 6-phosphate helps in the synthesis ribulose 5-phosphate, and then the ribose 5-phosphate into PRPP and purine pyrimidines are formed. Glucose 6-phosphate isomerase enzyme converts into fructose 6-phosphate. Fructose 6-phosphate directly takes it to the xylulose, directly to ribulose, and then the ribose. Similarly, fructose 6-phosphate after formation into fructose 1,6-bisphosphate. So this is glycolysis reaction. This reaction on the left is the glycolysis reaction. These are the intermediates. I hope you can see the cursor here. These are just the intermediates of the process of glycolysis. So these intermediates can kick into the formation of ribose 5-phosphate and purines and pyrimidines. Likewise, glyceraldehyde 3-phosphate that was produced in the process of glycolysis, and since 6-carbon glucose, this is 3 carbon, so 3 carbon glucose carbon compound means that 2 glyceraldehydes will produce. This 2 will eventually sorry, there's interruption. Kindly mute yourself.
Please mute. Thank you.
So glyceraldehyde 3-phosphate can go and form the ribose 5-phosphate. So these ribose 5-phosphate PRPPs and purine and pyrimidines form. So process of glycolysis is important. That's why I'm emphasizing again and again that the intermediates of the process of glycolysis are utilized everywhere in the body. Then this is fructose. Now, since we are doing carbohydrate metabolism, so the glucose was yielded in glycolysis. Likewise, glucose can be converted into fructose. Glucose 6-phosphate can be converted into fructose 6-phosphate. It is by the process by a simple enzyme, which is isomerase. Now, this fructose, if we take in fructose, so fructose is fruit sugar. Whenever we take fruits, it has fructose in it. So once the fruit is taken in, we get the readily fructose to us. So we are utilizing fructose. So one cycle where glucose is converted to fructose isomerase, that is skipped. The isomerase part is skipped because fructose directly enters into your glycolysis cycle. Fructose, instead of utilizing glucokinase or hexokinase, it utilizes fructokinase, and this converts fructose into fructose 1-phosphate. And then again, fructose 1-phosphate, if you can see the cursor, can go further by the presence of enzyme aldolase B. That's not aldolase S, it's aldolase B, and it helps in the conversion to glyceraldehyde or dihydroxyacetone phosphate. And again, the same reaction of glycolysis are the same. Fructose, glucose, after glucose comes fructose and glycolysis. If we take in fructose, it directly enters into glycolysis. Instead of utilizing glucose and hexokinase, it utilizing fructokinase. Also to mention, if you forget the name, it's kinase only. So you can write kinase enzyme. Kinase hai, if the fructose and converted into fructose 1-phosphate, it becomes fructokinase. It is glucose to glucose 1-phosphate becomes glucokinase. But then again, the importance comes where it is happening. If it's happening in the liver or it's being secreted by some of the pancreatic beta cell, then it's glucokinase. But if it's happening in the extrahepatic tissue, it becomes hexokinase. So kinase enzymes comes in, helps in the formation of fructose 1-phosphate. Now what happens if over here we don't have fructokinase enzyme? If fructokinase is not here, it will be it will not, we will not get fructose 1-phosphate. And this is leads to essential fructosuria. Fructose will start coming in urine. This is called essential fructosuria, which basically it's because of the lack of fructose. It is an autosomal recessive condition. It's a benign condition. It's not a problem. It's a benign one. And fructose accumulation takes place in the urine. We just have to counsel the patient to stop taking fruits and utilize the other means of energy. But what happens if fructose 1-phosphate cannot be converted to aldehyde? If there is no aldolase, if they have no aldolase, so what will happen? So if there's no aldolase, glyceraldehyde and DHAP will not take place. This is very important because aldolase is going to convert into glyceraldehyde and dihydroxyacetone phosphate, और यही glyceraldehyde pentose phosphate में भी हैं. This dihydroxytone phosphate is also in your ketone body formation, is also in your gluconeogenesis, it also goes into your glycogen synthesis, and it also one of the integral, this glyceraldehyde 3-phosphate is an integral part of your glycolysis process, because dihydroxytone phosphate converts back to glyceraldehyde so that the process of glycolysis can go forward. So if this aldolase is not present, then this is hereditary fructose intolerance. It's an autosomal recessive condition. Absence of aldolase B leads to intercellular trapping of fructose. So aldolase B agar come hai, so it's caused the trapping of fructose 1-phosphate, which because fructose is being trapped, it is not, it's this red line means it's not allowed. These further reactions are not allowed to take because it does not convert into glyceraldehyde 3-phosphate. So energy is being trapped. It's not converted into energy. This NADs and conversion. So this NAD is converted to NAD oxidizer. If it gets reduced, so फिर हमारे पास ATP produce होना शुरू हो जाता है. But if it's not utilized, so there is no energy. So patient is going to be severe hypoglycemic, leads to vomiting, jaundice, hemorrhage, hepatomegaly, can cause renal dysfunction, can hyperuricemia and lactic acidemia, because energy is not being produced, तो lactic acid body में produce होना शुरू हो जाएगा. Severe hypoglycemia होगा, vomiting होगी, lethargy होगी, renal dysfunction होंगे, because energy is required for the maintenance in all parts of our body. Now fructose, sucrose and sorbitol, they can all cause hepatic failure and death. We're going to discuss sorbitol later. And then the treatment is just removal of fructose and sucrose from diet, because most of the sucrose is also being extracted from the plant sources, from, for example, sugar cane hai ya hamare paas beetroot hai. So these fructose, fruit sugars and sucrose which are being extracted by them, they can be removed from the diet, and this will help in alleviation of this condition, which is hereditary fructose intolerance. इसके लिए याद रखें, the enzyme that is missing is aldolase B. Now other reactions. In other reactions, we have galactose. So milk sugar, galactose. So galactose is present. Now what happens to galactose? So this galactose, you can see UDP galactose can combine with glucose again. So glucose is very important. For all reactions in the body, glucose is very important. Glucose can easily be transferred. Transferase enzyme is here, where whenever something is transferred from one part to another, it's the transferase enzyme. So transferase, UDP galactose, the presence of transferase can cause UDP glucose and galactose is left behind. Similarly, UDP can be removed, and hence if galactose is there, you can have the lactose part. So this is beta galactose and this is glucose. So lactose हमारे पास इसका जो है बन जाएगा, इट्स डाइसैकेराइड लैक्टोज, ठीक है। नाउ galactose UDP, how this UDP galactose comes into being. So galactose, in the presence of galactose kinase, converts into galactose 1 phosphate. In the presence of transferase enzyme, galactose is converted into UDP galactose, and the rest of the reaction ये कि glucose के साथ combine करेगा, UDP will come out. Eventually galactose and glucose will combine together to form a disaccharide, which is lactose. So glucose and galactose makes lactose. This is it. But again, lactose tolerance है for those babies who do not have lactase enzyme, they are lactose tolerant, so they are given lactose free milk. फिर there are soy and there are other supplements which are available in the market that can also help in the removal of lactose from their diet. And once the lactose is removed from their diet, ample amount of source, ample amount of energy is provided to the other sources for their development. Then we come to this situation where there is uncontrolled diabetes. Now, up till now, glycolysis, glycogen synthesis, whatever the reactions we were talking about, it was all the normal situation where if the energy is required, then the rate limiting enzymes or the enzyme, they will start working so that more and more of the products is produced and more of the ATP is produced. What happens in a metabolic disorder, for example, in uncontrolled diabetes. So diabetes is a very common disorder where it is either qualitative or the quantitative deficiency of insulin. Now, if there's glucose is elevated, then glucose can enter into, for example, this is an example of liver, of kidney, lens, and uh, lens. So this is over here, the glucose enters into the cell. Either this glucose goes to the process of glycolysis, because glycolysis to cytoplasm mein hota hai, so glucose can go to glycolysis. If there is excess glucose, because this is a disorder, it's diabetes. So what happens if there is excess glucose? Now this excess glucose is converted in the presence of an enzyme aldose reductase. So this aldose sugar, glucose is aldose sugar, fructose is keto sugar. Aldose reductase, or you can say reductase enzyme, where this reduce NADPH and hydrogen, it is oxidized to NADP positive, and sorbitol is produced. So this sorbitol is osmotically active. Sorbitol is an osmotically active compound. It causes imbibition of water. Now, sorbitol cannot go out of the cell. Glucose can enter, go out of the cell, sorbitol cannot. It's an osmotically active. So once it's been trapped inside the cell, it causes osmosis. It causes higher osmolarity, and hence more and more water will enter into the cell. For example, this is the liver, this is the kidney cell, or this is lens. So in the eye, early cataracts develop in diabetes, and these early cataracts are because of the excess of sorbitol formation, because glucose, that much of glucose that is required for the maintenance of the lens, that much energy is produced by the process of glycolysis, while rest of the glucose in the presence of aldose